This is the third section focused on engineering, and we will start, I don't know if this is engineering or not, but we will start with a presentation from SwissDAO on Flux to Flow, Directed Evolution of Intent Allocation. That's a hell of a title. Take it away.
Thank you. So welcome. Today I want to walk you through two seemingly different domains and show how they actually converge. One of them being biology, and the other one, of course, being Web3. A little bit of background, so that you know where I'm coming from. I worked at Anoma, so I still work on this protocol. I have a PhD in systems biology, a master's in biotechnology, and in systems biology, and I worked on synthetic biology a while as a research fellow, where I worked on genome recoding and constraint-based optimization. This constraint-based optimization is something that comes in handy today, and I worked on metabolic modeling as well in one of my theses, and I'm going to draw some parallels between this metabolic modeling that I've done there and the intents on Anoma. So the motivation, the high-level idea is to check and compare the approaches and applications of some of these linear programming methods, which I'm going to refer to as SLP from here on out, and see how they translate from one context to the other. So we're going to look at the DeFi matchmaking domains, the analogies, and the potential pitfalls and gaps that are there. So what is an intent? This is the Anoma definition, an expression of an individual's desired end states. So it's a very declarative statement. In biology, we don't literally know what microbes want, but we kind of make an assumption on what they want, which is for bacteria, in this case, to grow, so to maximize their biomass. And this is thus, in a sense, a proxy intent in the Anoma language. If we look at this bacterium, Escherichia coli, which I have studied for too many years, here we see a metabolic pathway map, and all that I'm trying to convey here is that even though the cells are so tiny, this network is vast. It's absolutely incredible. It's about the size of 3,000 reactions that are.
Right, so it's reactions.
Yes.
So basically, there's going to be little lines between the little dots, and each of them is a reaction, like a chemical reaction, right?
Yes, these are metabolites connected by, the vertices are the reactions. And yes.
That diagram's amazing. Can I have it as a screensaver, please? Why are some green and some grey, please?
So this is the Kyoto map. It comprises all the known metabolic reactions. And I'm here focusing on this particular bacterium, so it doesn't have all the all the reactions that are in this database. So it's just a subset of them. And this bacterium it's a model organism. It's relatively tiny. But even if we look at only its central metabolism, then we see that the network is vast. And so this is only core metabolism. And if we zoom in even further to central carbon metabolism, then the network is still very, very large. So we see pentose phosphate pathway, citric acid cycle. You might know those from high school. The point here is that these kinds of networks, if we want to study them, they're incredibly difficult to study using differential equations, let's say. So what we do instead is we use an approach called flux balance analysis. In flux balance analysis, what we do is we take all these reactions that we know and we put them in a matrix. So effectively, it's a stoichiometric matrix of all the reactions with the metabolites on the one end and the reactions on the other. And there's a flux vector which describes the flux going through this network. And we make this assumption of a steady state, so mass balance. So the input equals the consumption equals the production. And this is obviously an assumption, but it allows us to make meaningful predictions. And it allows us to actually work with these very large networks, because now we don't need to know anything about these parameters that we generally don't have information on, or very sparsely so. So it's a massive simplification, but it works. So it's a method for predicting steady-state flux distributions in metabolic networks, and the rate, and what flux is the rate at which a reaction converts a particular substrate into a product. Metabolic model is there's a stoichiometric matrix, as I mentioned, with the metabolites and the reactions in there. All the stoichiometric coefficients are in there. This thing is mass balanced. And then there are gene-protein reaction rules in this model associated. So these are basically through Boolean expressions associated, meaning that when a particular gene either is there or is not there, we can turn, let's say, this reaction on or off. Then there are the flux bounds. So this flux factor we can constrain, and we typically do that, for example, using thermodynamics. Some reactions are considered to be irreversible, then we might put a lower bound of 0. We also can tune the nutrient uptake. This is just what is outside of the cell. We can simulate things like knockouts of reactions or gene knockouts by these associated gene protein reaction rules, and see what effect that has on the network. This is the canonical linear program formulation. So we try to maximize this flux, where we decide what the objective is. We assume that is biomass production. It could be to yield as much energy as possible. It could be a particular target flux that we want to produce if we're talking about metabolic strain engineering. And solving this yields us this flux vector that satisfies these constraints. So the space is initially unconstrained. So it's shown in 3D here, but it's obviously a high dimensional polytope. Then we impose these constraints. And then when we impose these constraints, we end up with this high dimensional polytope. And we can use linear programming after we define our objective function to find the optimal solution within this space. Now I want to kind of relate this to one of the examples from Anoma, which is the Kudos example, where we have two agents or two intents that are expressed here in the code. It's a bit tiny, but so be it. So there are weights associated. These two intents, they have they're haves and wants, and the haves, they have a value associated, which basically expresses how much the agent values that particular resource that it has. And now what I think is interesting is that we can effectively, so I've kind of done this exercise, and I should probably give this disclaimer, I'm not a mathematician, so my mathematical quotations might definitely be flawed somewhere. But what I think is interesting is that we can actually translate this into something akin to this stoichiometric matrix. So the wants and haves are, in that sense, just, they can be the positive values in the code over there, but we can conceive of them as being negative values when they're things that you want, and positive if you have them. So it's very similar in that sense. And now then there's the weights factor, which is various, which is the weights of the individual intents that are in this pool, and then the values, which are just how much these agents appreciate these particular resources that they have, so that when we try and optimize to find the optimal solution using linear programming, they get a, how do I express this properly? We find basically the optimal solution so that they retain as much of the resource as possible in the overall, in the system overall. And we also have the same thing with the conservation of flow that we also had in metabolic modeling that we saw, where we had the steady state assumption. Here also, the resources need to be balanced on both sides of the equation. So in that sense, these things are identical. Yes.
I may be using the terminology incorrectly. Is this field called linear programming?
Mm-hmm.
It is. So, with an intent, I might say that I want dinner, and I'm willing to pay 5 francs to get it. I don't necessarily want dinner twice, though. So I'm confused about this. And to be clear, this is not a hostile question. I'm trying to understand. Once I've had dinner, I don't want a second dinner. Whereas a bacterium, having divided once, because it increases biomass, to divide again. So that makes the bacteria's behavior linear. That I can understand there's a good match to linear programming. It's not immediately clear to me that there's a similar match for intents, where typically I might want something once, but then the marginal gain of having it again may be much lower. Do you have any comment on that?
Yes, I agree. There are definitely similarities here. There are also differences. For bacteria, we assume that they're growing in an exponential phase. So indeed, for the steady state assumption. And when we're talking about the intents in Anoma, this is a bit of a different story because we cannot assume the same thing. These intents are, in that sense, entirely different. They just also yield the same kind of a problem that needs to be solved. We're still talking about, well, flux in terms of the metabolic model, and we're talking about flow in terms of, well, when we're talking about settling intents, when we're trying to balance resources.
What you said to me is essentially, Jamie, yes, but it doesn't matter. You get the same mathematical structures out.
Precisely.
Thank you.
Yep.
It's not obvious to me, but you're saying it, so it must be true. Okay.
Well, let's hope. So yeah, there's some similarities. So this is the constraints that are there, the conservation constraints, the linear objective, both try to optimize this function. The flux balance analysis is pure, whereas the intense origin example from Anoma is mixed integer linear programming. These are slightly, basically the same. It doesn't really make a significant difference. The interesting differences are that, well, the intent is, well, explicit in terms of the agent expressing it, but in biology it's implicit. We, as a modeler, choose it. Then there's these binary decision variables that I just, well, it's basically the same point as over there that I already mentioned. So one of them is pure, the other one uses this mixed integer approach. And lastly, the routing scope is different. So in FBA, it's metabolized routing to a network, and with intents, it's resources via some global conservation that needs to be attained, that are flowing through a system of agents, or intents in this case. So then, building on this flux balance analysis, there's like loads of different approaches here, and I'm not gonna touch on all of them. I considered, but no. I'll touch on a few. One of them is flux variance analysis. So we do the same thing as before. We find the flux solution, optimal flux solution, and now we fix it, and then we try and minimize the fluxes with this fixed objective, with this fixed optimal solution in place. And then we see how much can these fluxes vary while still attaining the same optimal value. There's also parsimonious flux balance analysis, and what this tries to do is, again, we're trying to maximize this flux factor, this particular flux that we're interested in, biomass, for example, ATP or whatnot. But then at the same time, we also minimize the overall sum of fluxes in the system, which kind of expresses our desire to, or the organism's desire to not be wasteful. Let's say to use the minimal amount of enzymes, for example, necessary, because there can be very many paths in this network, but you want to kind of take the shortest path, kind of a way of thinking about it. So this also translates to one of the kudos examples, I think, quite well. So this is a bit of a different one. The kudos example here, there's this XOR kind of a logic that is expressed. So an agent doesn't necessarily want to, well, in the previous example, it had some resources and a particular preference for those, but here it's a bit more complex, or at least it's certainly different, in that the agent might not want to just get A or get A, but it might get either, might be willing to accept A or B. So that is the difference here. And it is very similar in terms of the framework, both optimize over flow. The conservation laws are again valid for both these, are in place for both these cases. And then there's this two-stage logic with flux variability analysis and the parsimonious version thereof, that we first fix the biomass if that's our objective, and then we try to refine the other fluxes. Whereas in the intent case, there's actually this or kind of a logic. So we first fix those and then we optimize the quantities. And then there's also some notable differences. Whereas one is continuous fluxes, there's again this mixed integer linear programming formulation on the intense part. The objective is slightly differently formulated, and the search space is definitely also very different because well, we are dealing with a convex polytope when we're talking about the metabolic network, whereas in this case for the intents we're dealing with a combinatorial XOR-based option set. Now, kind of the last method that I'll introduce is target flux. So here the idea is that if you have a particular flux distribution that you would like to attain, so let's say you're engineering an organism, or maybe there's a disease where fluxes are not according to what is considered to be healthy, and we want to try and intervene somehow, then we can try and take this idealized version compared with the current state of the network and see where we need to intervene, so to say. So which reactions should be upregulated or downregulated in the metabolic network. Then that allows us to shift, or show us which, shift from this current state towards the desired state. And why is it relevant to Web3? Well, we also kind of want to tune flows. It's kind of obvious perhaps, but balance liquidity to minimize slippage, to incentivize strategic pools and token adoption. So the target intent flows from real world data. We might see this desired pattern of a particular set of trades, let's say, and we want to steer the system towards this. So these become then our target flow, and we can, from using such a method, we can then infer which parameters we need to adjust in order to attain this. And here's a bit of a, well, it is a summary of the mapping. I didn't go into all of them. I only went to do the first two of these. I also did the other two. They're in the rest of the slides that I'm not going to present today. But yeah, there's, I think, very interesting similarities here. There's also some key differences among them. There's an even larger set of approaches that I mapped to Web3 protocols. But to close off, I want to say models are always approximate. So this flux balance analysis, variability analysis, etc., these provide us a best guess estimate of the flux distribution. But obviously, as always, and everywhere, we're dealing with incomplete information. So the predictions are, well, not perfect. And the key insight is that no in silico model can capture every variable, and biological systems with 3 markers are simply too complex for any linear program formulation to be truly optimal. So the takeaway from this is that this modeling is just a starting point. It's a design that we obtain, and it provides us something that we then need to iterate on. And this is effectively also how it's done in synthetic biology. We design things with an end goal in mind, but the system is so complex, it actually, more often than not, doesn't exactly do what we want. So we really just want to kind of move it to the right direction, let's say. If evolution takes over, then we can try and optimize for it. But we do need to put it in the right direction first, because if we just wait around until evolution comes up with it without us doing anything, that might take forever. So if we just push it in the right direction and then apply the right selective pressure, then we obtain the desired result. So the regular evolution in synthetic biology is this cycle of design, build, test, learn, same as in software engineering. So you propose a particular pathway or an enzyme based on particular predictions from one of these methods. You construct them, a library of mutants. Then you apply this selective pressure, so the cells grow, and you select for the ones that have the desired trait, and well, the others you get rid of. And this just, you iterate until you achieve something that is good enough, or that is as good as you can get. So the core principle being, by only applying selective pressure, we can navigate a vast design space and let evolution find solutions that models cannot predict. And in terms of, and here I want to go off on a bit of a tangent, I suppose, and see how this could be done in Web3 as well, such an approach, particularly in the context of intents. So we might imagine a set of evolvable parameters, a weight factor, the penalty coefficient, the slippage versus match priority, or these flow rule flags concerning full or partial fills, for example. We have certain metrics that we can define, like the match rate of our intents, the utility thereof, the unused resources, gas and latency. Then what we can do is create kind of subpools out of this with different variants of the linear programming. Route the new intents to random subpools. Then we can perform at the end of some epoch, 1,000 blocks or whatever, selection by computing the fitness thereof using one of those metrics or a combination thereof that I just suggested. And we can rank these pools and keep the best, drop the others. Now, we don't really want to flood the blockchain too much. So in biology, material also gets reused, and the blockchain things stick around forever. So we don't just want to spam new contracts all the time. So what we ideally do is we update the parameters of our contract so that in order to achieve this, yeah. And then we can emit these events for off-chain archival. The key benefits are that there's on-chain feedback. The metrics can be stored in the contract. We don't need any oracles. It can, it will be self-optimizing. The linear programming configuration adapts to user behavior. So you would have a self-adaptive contract or system, and that provides robustness as well. So when the environment changes, the protocol itself will naturally adapt. And with that, I have come to the end. I'll take any questions.
Thank you so much. Any questions from the live studio audience?
Thanks, very interesting. Have you explored other models that are used in synthetic biology as in, to see whether they apply to any like the Web3 and especially the intent business? Have you?
Yes, and I'm not going to go over all of that. Here is a small overview of such. It's a bit crammed, I suppose, but that's what you get. Yeah, but yeah, here are the methods listed, and then some Web3 counterparts, and some, and then I kind of try to express the relationship between these. It's not super detailed, but yeah, there are lots of these similarities. Lots of, yeah.
When will you write the journal paper on this?
No, what paper?
The journal paper.
The journal paper.
When you go, this deserves to be written up.
Yeah, I don't know. It could be interesting. Yeah, I'm no longer in academia, though. Thank God. So
there's an Anoma research topics archive that does accept submissions. Just
all
to
right.
let you know.
Well, I have to be honest. This has all been written up over the course of a day, so it definitely needs some refinement before I am willing to try and publish it anywhere. But yeah, no, I'm willing to iterate on this.
I just want to spell out here. There's real value in this table.
Yeah, I think so. And there's loads of more methods that I haven't gone into, but that are actually interesting to consider in terms of Web3 and particularly intents as well. Yeah, I don't want to just now go and click through all of them, but yeah, there's loads of them.
So is the observation that there's analogies here, but I mean, just in a very simple way, you're making claims here about things being in some sense the same, or at least
yes,
corresponding.
So in biology, we have come to the understanding that it's in metabolism, the important thing is flux. So it's not about per se how much of a thing you have at a given point, but it's like the rate at which things flow that matters, that it's significantly steering a system. And I would say that in Web3, that's the same thing. It's not about people hoarding their assets. That is
People
hoarding their assets. It's about people actually using them. It's about the flow. Protocols want flow.
That needs to be in your abstract. That's.
One more audience question.
Thank you.
May I point out the elephant in the room?
Please.
You mentioned it in passing, though. This is all equilibrium stuff. So large number of things, and by and large predictable. Whereas stock markets typically have like an element of stochasticity. So, and just as a remark, there are stochastic models, as in particular that I was asking for, and yeah. So in particular, one of them I will not mention. The other one is like work for Peter Stadler on autocatalytic cycles. So basically, the idea is that on the one hand, yes, you're interested in the quantitative side of things. On the other hand, you can also just look at the structure of intents and try to find what would be in biology called an autocatalytic cycle, meaning that basically you do a bunch of things and you go back to where you are, but with additional liquidity. And that would be definitely interesting if we can find any of those structures and intents. And I would also,
I agree.
I would like to keep.
No,
up the
I think this could be a super interesting area of research. Yes, I think there's lots of similarities there, and well, it's a huge simplification, of course, the steady state assumption, but it actually provides us very useful predictions in biology, and I believe it will be able to do the same in larger systems outside of biology as well. Yeah, it's really about the scale that you're operating at. When we are talking about modeling, you need to approximate things, you need to make simplifying assumptions, and then, of course, thus iterate on those, maybe evolutionarily optimize them. But yeah, if you want to do this for these very vast networks, which we're also dealing with in Web3, there's no way to try and describe these dynamics like accurately. That's just impossible. And I would argue you can try a little bit, you can use some approximations, but like what would such a system of equations even look like? Like too many parameters, this is not going to be useful, in my opinion. maybe some super good neural network will figure it out in the future. We'll see.
Okay, challenge accepted.
All right, well, if you came here for engineering, we just forgot the word biology in front. Let's give it up again.